Hello and welcome to this lesson recording on gene mutations. In previous lessons, we've looked at the structure of DNA and RNA. We've spoken about DNA replication. We've spoken about transcription. We've spoken about translation. And today we're going to be talking about how gene mutations can utilize all these processes in order to cause changes in an organism. So by the end of today's lesson, you should be able to describe the basic types of gene mutation. You should be able to explain the consequences of a gene mutation, and you should be able to evaluate the effect of mutagenic agents. So let's start by using a keyword. All keywords are in green. We're going to define the word gene, okay? A gene is defined as a small piece of genetic material that codes for a characteristic. That codes for a characteristic. Now, when we say a characteristic, we've said in previous lessons that you've got these base sequences inside of DNA, which are converted into mRNA-based sequences, which are then read by the ribosome to make proteins. The proteins give us our characteristics. So I'm going to put in red underneath there, because again, it's an important point. We can say that genes code for proteins that give us characteristics. Okay? So it's these proteins that give us the characteristics that we have. So for example, if you have dark skin, your genes code to give you lots of the protein melanin, which makes your skin darker, okay, as a protection against UV light. If you're albino, for example, you don't produce as much melanin, therefore you lack the gene to produce that melanin. There is not as much transcription and translation of the gene that produces melanin to give you darker skin. Instead, you have very pale skin. Now, leading on from this, we've got another key term, the term gene mutation. Now, a gene mutation can be defined as a change in the base or the nucleotide base sequence of a gene. Okay? Now, the human genome has about 3 billion base pairs, and just one change can cause the protein made to be different. So we can say, I'm going to put it in red again, that the human genome, and we don't spell human genome with an I, do we? The human genome has about 3 billion base pairs, and a change to just one of these bases can alter proteins made. Can alter proteins made. Now, these changes of a base can usually happen during DNA replication. So these bases can be changed. DNA polymerase can make a mistake when doing DNA replication, and it can change one of the bases involved. So we can say that changes to bases occur as errors in DNA replication. Okay, so during DNA replication, DNA polymerase can make a big mistake, and it can cause one of the bases to be different as a result. So what we can do now is we can have a think about how this can affect the processes of transcription and translation. So if we've got an original piece of DNA, so I'm going to put original DNA, and we're going to start with TAC, because TAC will always code for a start codon in an mRNA molecule. So we've got TAC, and then we're going to go G, 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 then we're going to go A, C, C, then we're going to go A, T, G, and I think that'll do. We've got what? 1, 2, 3, 4 codons. Now, if we've got a mutated piece of DNA, we're going to change one of these. So we're still going to start with TAC. We're then going to have CGG GGG as we had as normal. Then this time, instead of an A, we're going to put a C. So C, 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 ATG. So our mutation has happened here. Okay? Because in this one, in the original DNA, it's an adenine. As opposed to in the muted DNA, there's been an error during DNA replication to turn this adenine into a cytosine. Now, if we think about this in a step-by-step process, this will change the molecules being made. So, for example, our original mRNA that would be produced from this DNA would be A, U, G. It would then be C, C, C. It would be then U, C, C, U, A, C. Okay? In our mutated DNA, it would be, so we've got our mutated mRNA. We would still have AUG. I've picked that codon for a specific reason. We would still have CCC. However, now, instead of a U, we would have a G. Okay? And then we can go CC, U, A, C. Okay? We've still got our mutation. Instead of having a uracil in our mRNA, we're now going to have a guanine. Now we can break that up, as we've said in previous lessons, into our codons. So we've got codon 1, codon 2, codon 3 and codon 4. I'm just going to put C1, C2, C3 and C4. That stands for codon 1, codon 2, codon 3, codon 4. If you wish in your notes to actually write out codon, please feel free. Again, over here, we've got codon 1, codon 2, codon 3, and codon 4. So codon 1, codon 2, codon 3, and codon 4. Now, these first two codons are identical, and so is the fourth codon. They are identical. Okay? But we now need to think about the protein being formed. So I'll switch to this yellow colour, and we'll put original protein. And I've literally made this up as I go along. But the first protein we've got from our mRNA during transcription, so we've had transcription, we're now going to have translation, and I've prepared this earlier, is AUG. So we want our mRNA amino acid table. See what comes up. So we've got AUG. This would be leucine. So I've gone from the first letter A, second letter U, third letter G, leucine, LEU. So AUG in both of these proteins would code for leucine, LEU, and then over here we would also have LEU. We would have leucine. So this is our mutated protein. Our next one is CCC. So C, C, C is serine, SER. So our second protein for both of these would be serine, SER, SER. Now this is the moment of truth, because I haven't pre-planned this. I've literally done it off the top of my head. It may fall flat on its face. But our third codon for this one is UCC. So UCC is serine. So for this first one, we're going to have a second serine attached to this, SER, whereas this one, GCC, GCC is alanine. So just because of this single base that's changed, we've now got a different amino acid in the chain. Our final one is UAC. UAC is tyrosine, TYR. So both would still have TYR. And that would be our polypeptide formed. Okay, that would be the primary structure of the two proteins formed. Now, because serine and alanine would have different R groups coming off them, this protein would fold in one way due to its tertiary structure. The R groups on serine would cause this chain to fold in one way, whilst the R groups on this alanine would cause this protein to fold in a different way. Therefore, you've got two differently folded proteins which will affect how it works. You've got a mutated protein which may no longer be functional or may do a totally different job, or you've got a normal protein doing the job that it wants. So we can just add underneath here, I'm going to put in regs, it's important, that the mutated, and I've just had a thought that I am recording, the mutated amino acid alanine, isn't it? Has a different R group to the original, which may make it fold different, may make the protein's tertiary structure, different. So we've said that a gene is a small piece of genetic material that codes for a characteristic. It does this by coding for proteins, building amino acids in translation, using mRNA made in transcription. The gene mutation is when there's a change in the base sequence of the nucleotides. The human genome is about 3 billion base pairs, and a change in just one of these bases can alter the protein made. Changes to bases occur as a result of DNA replication. So you may have an original piece of DNA that looks like this. Due to an error made in DNA replication by DNA polymerase, instead of having an A here, you have a C. What this means is that the original mRNA has a U at this position, but it now has a G at this one. And as a result, the original protein that was intended to be made was leucine, serine, serine, tyrosine. Instead, in the mutated one, we've now got leucine, serine, alanine, and tyrosine. This would mean that there is a different R group attached to this amino acid, and therefore the protein chain or the primary structure of the protein will be different, which may lead to a change in the tertiary structure of the protein. Now, there are different types of gene mutation. So this is a recording. You can pause the video at any point if you haven't got all this down. For now, I am going to clear the screen so that we can go on to the next bit. I'm also going to have a swig of water. Apologies if this comes down the mic. So there are different types of gene mutation. So there are various types of gene mutation. First of all, there are two broad groups of gene mutation. Okay, there is a silent gene mutation and a non-silent gene mutation. So we can say, and I'm going to put this in red because it's important, there are two groups of gene mutation. We've got silent gene mutations and a silent gene mutation does not change the protein produced. So in a silent gene mutation, the protein produced is the same as the original. And we've also got what's called a non-silent gene mutation, where the protein is not the same. So non-silent gene mutation. And we can say that the mutated protein is different to the original. Different to the original. Okay. Now, if we're thinking about some other different types of mutation, we've got what's known as an addition mutation. an addition mutation. Now this is where an extra nucleotide is inserted, so there is an extra base added to the sequence. So addition mutation, we can say an extra nucleotide is added to the sequence. Okay, an extra nucleotide is added to the sequence. So let's say rather than having A, T, C, G, G, you're now going to have AAT, then we're going to put in a G, and then CGG. Now, you know that it's an addition mutation. A quick way of working this out is by counting the number of bases. So, for example, this one started with 1, 2, 3, 4, 5, 6. It's now got 7. 1, 2, 3, 4, 5, 6, 7. So you know something's been added. Okay. On the other hand, we've also got a deletion mutation. This is where a nucleotide is removed from the base sequence. A nucleotide is removed from the sequence. So go back to our original. We've got A, T, C, G, G. What will happen is we'll lose one of these. So rather than having AAT, you would have A, T, C, G, G. An easy way to work that out is count the number of bases. There's one less. You've had a deletion. Another example would be a substitution. Mutation. A substitution mutation is where one nucleotide is swapped for another with a different base. So where a nucleotide is swapped for another with a different base. Now, a substitution mutation is what we were talking about earlier. So rather than having A, T, C, G, G, you may have A, T, C, G, and then an A. So we've swapped this G for the A. Okay. That is different to an addition mutation, as we're not actually adding a base in an addition mutation. Okay. Sorry, we are adding a base in addition mutation. In a substitution, we're swapping one for the other. Final couple we're going to talk about, however, there are lots of these. An inversion mutation. Inversion mutation. An inversion mutation is where a part of the sequence is placed in reverse. So where a part of the sequence is placed in reverse. So rather than having AAT, CGG, you may now have AAT, GGC. So we've swapped it around, it's inverted, okay? The final bit is a duplication mutation. So a duplication mutation. And we can say with a duplication mutation, the sequence is placed in twice. So we can say where part of the sequence is placed in twice. So rather than just having AAT, CGG, we've now got AAT. There's a repetition of AAT, and then we're going back to CGG. Okay? So these are all some examples of different types of mutation. You can have a silent gene mutation where the protein produced is the same and that has absolutely no impact on the organism whatsoever. Its characteristics are the same. You can only tell by looking at its genotype. Or you can have a non-silent gene mutation which changes the proteins made and would ultimately have an effect on the characteristics of the organism. There are addition mutations where an extra nucleotide is added to the sequence. There are deletion mutations where a nucleotide is removed from the sequence. Substitution mutations where a nucleotide is swapped for another and a different base. Where a part of the sequence is placed in reverse, inversion mutations. And duplication mutations where a part of the sequence is placed in twice. Now, on my PowerPoint, I've got a perfect analogy for this. So I want you to imagine an original, just a sentence. It says, the old men saw the lad. Now, if you think about it, these represent codons because they represent 3 letters each. So it's quite a good analogy. So the old men saw the lad. If we go an addition mutation and we add a C onto the old, it would now read, the cold men saw the lad. We've changed the meaning of the sentence, much like we could change the amino acids in the protein. A deletion mutation, the old men saw the ad, so rather than seeing a person, they're now seeing an advertisement, again, changing the meaning of the sentence. A substitution, the old hen saw the lad, so we've now got rid of the old men and we're now using an old chicken of some sort, and it's seeing a person, but we've changed the meaning from the original. An inversion, the old men was the lad. So the old men were the lad as opposed to seeing the lad. We've changed from the original due to inverting the 3 letters in saw. And finally, duplication, the old men saw the lad. So we're talking about very old men now, implying that they're, 100 years old. So you can see that just by changing these letters or adding letters, removing them, substituting them, inverting them or duplicating them, we're changing the original meaning. And this is exactly what happens when a ribosome tries to read these proteins. So if we have a final think about the consequences of gene mutation, so if we can put the subheading please, the consequences of mutation, the consequences of mutation, and we've said earlier in the lesson, I demonstrated this at the beginning, there are different polypeptide or a different primary structure can lead to a protein that folds differently. Okay, so we can say that different polypeptide chains may have different amino acids with different R groups, full stop. And we can say that the R groups may alter the tertiary structure of the protein. Essentially, we're saying that the polypeptide chain folds differently, and if it folds differently, it may not have the same properties. So we can say if the protein folds differently, comma, it may not have the same properties of the original protein and work differently. And work differently. Okay. Now, when these mutated cells duplicate, they pass on this mutation to their daughter cells. So if a cell has got a mutation and then it goes through mitosis, it will pass on this mutation to its daughter cells. So we can say that mutated cells pass on the mutation to daughter cells during cell division. Now, if this mutation is inside of a gamete, so inside of a sperm or an egg, this mutation can be passed on to offspring. So we can say, I'm going to put it in red because it's important, that if the mutation in gametes can lead to offspring with the mutation. Okay. Now, sometimes the effects of a mutation are so damaging that it can't be passed on. Okay. So, for example, if you have a mutation in the genes that code for, I'm just making this up off the top of my head, the formation of the alveoli in your lungs. If you do not have the proper formation of the alveoli in your lungs, you are far less likely to survive, and therefore you are far less likely to have offspring, so the mutation cannot be passed on. So we can just say over here that oftentimes the effects of a mutation are so damaging that the organism does not survive long enough, to pass it on to offspring. Okay? So let's have a think about some mutations that can take place. So one example would be during the formation of tumours, the formation of a cancerous sort of growth. So normally what you would have is a gene inside of cells known as a tumour suppressor gene, which would inhibit cell division. So eventually cells would go through mitosis until you no longer need to grow, repair. Therefore, the cells around that cell will send out this inhibiting protein, which stops cells around them undergoing mitosis. On the other hand, if you have a mutation in this suppressor gene, you can no longer send out this signal, this protein that stops other cells going through mitosis, and therefore these cells will go through uncontrolled mitosis and they will form what we call a tumor. Okay? Now, mutagenic agents, a mutagenic agent can be defined as a chemical or radiation which causes a mutation. So a chemical or form of radiation that causes a gene mutation. So mutagenic agents cause mutation inside organisms. It doesn't necessarily cause cancer because those would be known as carcinogens, but a substance can be a mutagenic agent and a carcinogen. However, a substance can also be just a mutagenic agent. We can cause these mutations without causing cancer all of the time. Some mutations are useful. So, for example, the albino thrush, which is shown on the screen at the moment, it has a gene mutation that stops it making a black pigment. So the one shown on the left has this black pigment inside of it, whereas the albino thrush on the right doesn't. It is clearly white. Now, because the albino thrush is all white, it has a very poor chance of survival. Okay? Now, if it had these dark spots on it, it would be able to stand on the ground, it would be able to be more blended in with its surroundings and less likely to be eaten by a predator. But the mutation it's had, although it's not cancerous, it doesn't aid its survival. Sometimes it can happen the other way around. Sometimes you can have organisms that have a mutation that make them more camouflaged or make them more likely to survive in their environment. Okay, that's the basis of natural selection. However, I just wanted to point out here that mutations are not always bad. People seem to associate mutations solely with cancer, and while it certainly is a big part of cancer, it's not the be-all and end-all that it only causes cancer. It can also be a beneficial mutation. So, you should now be able to describe the basic types of gene mutation, you should be able to explain the consequences of a gene mutation, and you should be able to evaluate the effect of mutagenic agents. Thank you for listening. If you have any questions, please send me an email or comment in Google Classrooms, and I will get back to you as soon as I can. Thank you very much.